Welcome to the Nucleic Acids and Proteins section of the Macromolecules of Life. This should come after the carbohydrates and the lipids section. So I think that it's really a pleasure to teach you about nucleic acids and proteins because this is sort of the really exciting part of biology these days as we are flying into the age of genetics at an incredible pace. So genetics, of course, is all based on genes that are held in the DNA and the expression of those genes, which makes proteins. So this is really pretty awesome. The diagram that you have before you right here shows you a few things that are here. One is the structure of DNA in the middle here, which is that double helix. And the next is the structure of RNA, which is different than DNA because the sugars are slightly different, but more importantly, it is a single-stranded molecule. The average length of DNA is in the millions of bases. So DNA can be up to something like, 100 million bases long. Enormous molecule, very, very stable. And RNA, the average length of RNA is going to be only about 1,500 bases. So it's a lot smaller. It's also, it lasts typically about 72 hours. If we're talking about messenger RNA, which is one of the types of RNA that we're dealing with. And so it's really a very tiny, tiny molecule in comparison to DNA, and it's much less stable. So let's go and talk a little bit about the structure of these. I guess where we're going to go today is we're going to start with the DNA, and from DNA we're going to produce, through a process called transcription, we're going to produce RNA or messenger RNA, and from messenger RNA we will produce, through a process called translation, will produce proteins. Proteins are a string of amino acids. It's based, I call it a string, but it's a polypeptide, a polymer made of a series of amino acids. So let's go in and look more closely right now at the structure of DNA and talk about that. Also on the list that you have here are these nitrogenous the nucleobases, which I will, of DNA and of RNA, which we'll get to in just a moment. At first, if you were to flatten out of the DNA, it would look something like this. It would be, I would have two strands like this, and you'd have a ladder-like structure like this. And we call these two things on the sides the backbones. And the backbones are made of a sugar and a phosphate, sugar-phosphate backbone. So and it sort of looks like a ladder. And then the rungs of the ladder are what we call nitrogenous bases, but you might have a pair of them like this, A with T and G with C, and these would be called the bases. And an entire chunk of this stuff that goes together, like part of the backbone and the base together, is called a nucleotide. Because that's sort of how it's assembled. Each little nucleotide comes with a piece of the backbone and a piece of the nitrogenous bases in the middle. So let's take a closer look at the actual structure of that. Now DNA is called deoxyribonucleic acid, and that's what DNA is short for. And if you want to do it in Spanish and have a good time, you can remember that in Spanish the adjectives come after the nouns, which makes the pronunciation of it a little different. So it's ácido desoxyribonucleico, which is kind of amusing. I'm going to draw this right like this. I mean, you're going to see me making this. I have a trick pattern that I use for drawing it. And there it goes. These are the phosphates out here, the circles, and these are the pentose sugars that are five-sided sugars. They go right there, and I'll make one more of these. And it's really a lot easier if you draw it out without the linkages first. This is one side of the backbone. The other side goes the other direction. So this is a little bit tricky, which means that I'm going to make these little pentose houses pointing arrows the other way. So we call this the 5' to 3' or 3' to 5'. The point is there's a directionality here, which means that the molecule actually is pointed in one direction on one side and in the other direction on the other. Then I can go in and put in my phosphates out here, and I'm going to link them together in the same way, but this time just going this way. Okay. Now, in the middle then of this thing, so this is again the sugar phosphate backbone. This is sugar, and this is phosphate. Don't you love this? This is the meat and potatoes of DNA. And now I'm going to draw the nucleotides that go in the middle. And I'm going to use the model of this drawing. I really love this image here. You'll notice that it has, you'll see the 4 of them here. Thymine is the T, and cytosine is the C, and adenine is the A, and the guanine is the G, ATCG. And we know that A pairs with T always, and that C pairs with G always. So that's the pairing of these. So if you want to get a really cool tattoo, you can get the tattoo of the ATCG so you never forget it. But I think that's kind of fun. So what I did was I drew out some little model structures on the side here to make look like these things. So the TC I think of as the small ones, so teeny weeny TC if you want. And then the other ones, the A and the G, are kind of awkwardly big if you want. And so I made the, let's say the first pair is going to look like this. I'm going to put a little house over here. It's got a short house. And that means on the other side I'm going to have a long house with an inverted section that fits it at the top, right? And then on the next piece down, I'm going to make the other kind of house, which is this kind of house with an M on the top. That's my cytosine, and that's going to pair only with the guanine that has a house that looks like it fits in there, okay? So this is a G, and this is a C, and this is an T, and this is an A. Now, by the way that I've drawn these structures, you can see there's only one way they can fit together. If you flip the A over and put it on the other side like this, it can still only pair with a T shape on the other side. Otherwise, the pairing is too wide or too small, and it doesn't fit between the sugar phosphate backbone. So this is an A and this is a T. All right, so you can see from this, I guess that I really love this part of the drawing where everything fits. Where the width here is exactly the same across here, right? And these things are actually part of they're bound by this. There's another one here, another one here. So this whole structure is called a nucleotide. So if I were to take a blue line and just draw around this, so you get a piece of the base, a piece of the sugar and the phosphate. Sugar, phosphate, and a base all go together to make a nucleotide, so I'll label that. Now, so that makes it inevitable that if you have one side of this, as you can see, you can easily fill in the other side. So if you, oh, I almost forgot the ever important, I wanted to put hydrogen bonds in here. In the middle in here are little hydrogen bonds, so we can use those from our lecture on the structure. These are H bonds. And there's some water in there to make those happen. How exactly they work, they do stabilize DNA, and so that's one of the reasons why it's such a stable molecule. So this goes on and on for millions of bases, right? This is really long, and that's DNA. Now you can see from this molecule that it would be very easy to fill in one half. Let's say that you cut the molecule down the middle using some sort of a cutting device. So I'll take out these little width markers here, and I'll just say let's cut it right down the middle. if you only had half, and your half said, for example, it said TCA, you could easily recognize the only thing that fits in there is AGT on the other side. This suggests that DNA is easily replicated. That is, that once it is unzipped down the middle, it's easy to fill in the complementary bases on the other side. So when you do that, if you had AGT on this side, you would fill that in with TCA, and of course now you have two identical strands. So when DNA replicates, it's very replication is what we have high fidelity, has high fidelity, and that means that it makes very few mistakes. It makes mistakes about 1 in a million. 1 million bases is a mistake. So if you think about, for example, if you run a zipper, how many zipper tongs can zipper pieces can you zip before you have an accident and you actually zip in one that's the wrong shape or doesn't fit? And that happens about 1 in a million times in DNA replication, and we call that a mutation. All right. However, it's very rare, but it does happen. Since you have billions of bases, eventually it does happen. Now let's look at the structure of that DNA on a larger scale. That DNA double helix like this, as you see in this image, this is the double helix. It is a fixed distance between the two strands all the way across there. It's pretty cool. And then those things are compressed and wound up around tiny little proteins called histones that are kind of like winding up thread on spools until it compresses into this large thing down here that is a chromosome. So that chromosome that you would see is actually a long DNA molecule all wound up really tight. Okay? So that is the structure of DNA. If we were to look at the structure of RNA, RNA is very, very similar to DNA, except it's single-stranded. And instead of having a T for thymine, it has a uracil. So you see that big uracil up there. And so it has the same shape as the T. So I would say that uracil has also this little house shape like a T, and that's the U. And so other than that, RNA is very, very similar to DNA. So let's take a look at where we sit. We've looked at this DNA here and its structure, and now we're going to look at how RNA gets made from DNA. And the way RNA gets made from DNA is shown in this little picture here. If you split open the DNA, and you can see it's split open here, and it's split open down here, and then in there you slide this thing that is described in here as polymerase, right? Polymerase, RNA polymerase, and what it does is fills in complements of one side. So you can see it's making this strand here. Right here is where it's actually filling in the complements, right? And you can see we've opened up DNA, and now we're making this strand here. And this will be continued to about 1,500 bases long on average, maybe longer, maybe shorter. And that is a strand of messenger RNA. Now notice that the U's in here have replaced the T's, but they are now pairing with the A's. And so I think about this as me running visually. I think I have a potato peeler. I have a great big Idaho potato, and I've got my potato peeler, and I put it on the potato, and I start moving across the skin. And as the skin peels off, basically what I'm doing is peeling off a large RNA off and reading it off the potato, if you will. It's sort of like that. Once the polymerase has passed, DNA collapses back together, and that is how a messenger RNA reads a section of DNA. Now that section of DNA, if this is DNA, you have to use your imagination here. I'm going to add a little bit of genetic information here. We might say that a certain region of DNA is called a gene. A gene is a region of DNA that codes for a protein. So what I'm going to tell you is that this is the code of life, right? So that little strand there that might look like A, T, A, C, 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 G, G, A, T, G, A, G, G, C, A, and so on. That is the gene, right? And let's say this is a gene for something like keratin. I like keratin because it makes fingernails. It also makes hair, and you'll learn about it a little bit when you do the epidermal lab. Fingernails and hair is a protein that makes those things. Now let's imagine that somewhere in your DNA you have a region that codes for keratin, and what we would look for is how does that gene get expressed. And the way that it gets expressed is you have, from that sequence, you read a sequence of messenger RNA is transcribed, right, like this picture, right? And then from messenger RNA, the sequence of amino acids is what we call translated. At the ribosome. So what you should be able to do for this class is take a given strand of DNA that I give you that might look like this, one side and the other side. It might say A, T, T, A, G, C, C, A. And on the other side of the DNA it says T, A, T, C, G, G, T, and you might have to fill that in. And then I might say this is a gene, and I'd say what would be the mRNA strand? What would it look like? Well, you'd have to fill that in here. That would be red off of that one, right? So what would that look like if you read that gene with the RNA polymerase? It would be the complement of A. So the complement of A would be T, but T is replaced by U. So it would be U, A, U, G would pair with a C, C would pair with a G, G, A would pair with U again, okay? So this is the mRNA strand. Yay! So that's pretty easy to do, right? Now that you have the mRNA strand, to translate that to protein sequence is done like this. Every 3 bases, like this, every 3 mRNA bases codes for a protein, for an amino acid, not just a protein. Okay, a protein itself is made up of a string of amino acids, so we sometimes draw a protein like this. And each of these is called an amino acid, right? Amino acid, AA for amino acid, and so on. Now that string, if you have a 500 base long messenger RNA, that string would be 1500 mRNA divided by 3 equals 500 amino acids, right? So the average protein is about 500 amino acids long. All right, now what are these amino acids? There are only 20 kinds of amino acids in the human body, okay? And I made a little map. There's a little chart of them right here, if you can look. This chart shows you how the translation goes. So if you have a U in this first box, you make the phenylalanine, which is amino acid. You can pick, if you have GAG, GAG, that's glutamate, right? If you have 3 A's in a row, that's lysine. So these are the ways they get translated, and the ribosome translates it, all right? And the ribosome does it with the help of a thing called transfer RNA. And that's another kind of RNA. tRNA does the translation I'm not going to explain that as part of this class. It's just tons of videos online. You can see it, but it does it with the ribosome, which is actually itself made of RNA, but it's a very much larger chunk of RNA that does this whole translation part. So essentially we're going to read now for each of these. Is going to make this one, this 3 here, we'll make this one, this 3 here, let's say we get another C on there, we'll make this one. And so if it's UAA, I can go look up UAA and see what it becomes, and UAA might have turned out to be a terrible choice. Let's see, there's UU. Uh-oh, UAA is a stop codon. That's not good. So it really couldn't be, and it doesn't code for an amino acid. So bad choice on my behalf. But you could see stop means stop translating and end the protein, so we can end before we even begin. But let's try UCG, the next one. So UCG is serine. So what I can see from that is that serine, this one right here, I chose these at random, actually codes for serine, and so this would be serine in the sequence here. All right, so that's serine, and that is how you get from DNA to RNA to protein. This process here, where we go from DNA to RNA to protein, like this, is called the central dogma of protein synthesis. And isn't that awesome? So this is a one-way street. We always start with DNA, we always go to RNA, and then we go to protein. And you can call this messenger RNA if you want to make it specific. But messenger RNA to protein. This is how all the proteins in your body are made. If you have a protein, it gets made by coming from DNA to RNA and protein. Now you also know what a gene is. So if you go back here, we remember that a gene is this coding region of DNA, right? So it's just a series of A, T, C's, and G's in a row that, when transcribed to messenger RNA and then translated to amino acids, yields a sequence of amino acids that makes a protein that then has some function like keratin in your hair. That's pretty awesome. Let's take another close look for just a moment at the structure of the protein itself. Now, proteins have a nitrogenous backbone. So the backbone of the protein goes like this. So let's look at protein structure for a second. It goes N, C, nitrogen, carbon, carbon, nitrogen, carbon, carbon, nitrogen, carbon, carbon, NCC, 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 NCC. So that's the backbone, right? So it has a nitrogenous backbone. And then for each amino acid, they all have in common, you see this nice diagram here. This is why it's called an amino acid, right? Because this is the base amino acid. It has an amine group and an acid group. But more importantly, it has this R group. Now the R group is what makes each of the 20 amino acids unique. So you can pretty much tag on here an R group. Now the R groups are going to have different shapes. Some of them will be long and skinny. Some of them will be round and chunky. And they're all going to have like different charges and different shapes. So when you make this long string of pearls, if you will, each of them has a different R group on the outside. So when you allow them to sort of, that snake of pearls each has all these R groups on it, it then collapses on itself and can make these unusual shapes as a result of the bonding of those R groups. So the R groups cause the shape. Now, then it gets a little bit complicated. So let's take a look and see if I have any other images in here. I don't. Looks like we're going to go to the PowerPoint and take a couple looks in there at what proteins look like when they fold together. Remember, there can be hundreds and hundreds of these little, like this amino acid, there's hundreds and hundreds of them in a protein. And proteins do a lot of things, including forming the structure for things like hair and skin, and they also are the little motors that move muscles. So they're very, very important.